Thank you so much for inviting me to speak today. I've been to past WAGR Weekends in person, and they were truly the most fun events, and my family enjoyed it so much too. So I hope that once this whole COVID situation resolves, that we can get together in person soon. So this is a update on the talk that I gave at the PCORI meeting 2 years ago, and I distilled it down to 12 minutes, hopefully, of the key parts that I think would be helpful. So the subtitle for my talk is actually, if I can, oops, how do I use next screen? Sorry, I'm still learning how to do this. So slide advancement, what do you think? Is it, here we go. Okay, so I can use the dial. All right, so I subtitled this Families and Researchers Working Together to Improve Clinical Outcomes as How to Train Your Dragon. And the reason is because these movies and TV program are my family's, one of our favorites. And I think it's because it really talks a lot about how when working together, there's synergy and you can do so much more by working together. And so I thought that would be a fitting subtitle for my talk. I would like to share the work that was conducted between the years of 2016 and about 2014 at the NIH, and then continued after I left and went to Memphis, Tennessee. And the focus really is on what we found there and kind of the future directions that we'd be going because of the research. So the concern that I had when I was a fellow was to pick a research topic in which we could learn about genetic disorders and use that to design new treatments to help people with rare disorders, as well as using rare disorders as an example to treat more common conditions that are in the general population. And so WAGR syndrome, despite it being very rare, can teach us so much about genes that are really important just for the rest of the population. And so I think that we can all work together to improve health for all people through our research studies. As you all know, the WAGR is an acronym. The W is for Wilms tumor, A for aniridia, G for GU anomalies, and R for range of developmental delays. And the cause of this is heterozygous deletions on chromosome 11. So everybody has 2 copies of genes, one from mom, one from dad. And so if one of the 2 copies is affected, then you have heterozygous, meaning one of 2 or have the deletion. And the cause for the Wilms tumor and the GU anomalies is a loss of 1 copy of WT1. Aniridia is caused by loss of Pac6. But at the time when I started studying WAGR syndrome back in 2006, we really didn't know what the cause of the range of developmental delays are. What we did know, though, was that there are some additional features when children have bigger deletions that include a gene called EXT2 that they can get osteochondromas, which are tumors of the bone. And furthermore, if it's an even bigger deletion and it has ALX4 deleted, then you get holes in the skull called parietal foramina. And so kind of looking in the other direction, there's a gene that lives up here, which we kind of wondered, maybe this gene, BDNF, brain-derived neurotrophic factor, might be the gene that causes the excess hunger and obesity that is seen in some, but not all patients, as well as impaired pain perceptions. There's really high pain tolerance that some of the parents were reporting in children. And why is it that BDNF was kind of on our radar? Well, the reason is because about a decade earlier, so back in the 1990s, people had looked at mice that had BDNF deletion. And so this is a BDNF mouse that has 1 copy of BDNF deleted, and then a wild type litter mate, meaning that it's a normal genetic type brother. And you can see there's a big difference in body weight. And so there was overeating or hyperphagia in the mice, as well as obesity. And then these mice also had reduced sensitivity to pain, and they had impairments in their social interactions, learning, and memory. And so because of this cluster of symptoms the mice were showing, it made us wonder if perhaps this might be important in people, that maybe it's in people also the BDNF deletion is causing obesity and neurocognitive impairments. So to study this, we conducted a study at the National Institutes of Health. And our main focus was on BDNF, but we also figured since people are coming, we should look at everything else too. We should look at all the other genes that are in this region and see if there are additional phenotype, which means the symptoms, genotype, which means which genes are deleted, correlation in this region. And so in order to do this, I am so touched and grateful by the time, effort, and sacrifice that families and patients donate to us by coming to the NIH. And you can see that the rigorous schedule that they had to go through for an entire week, it was just packed and packed with testing that they had to do. And so I just can't even, words just cannot even express how grateful I am that for the sake of improving medicine and research knowledge that families gave of themselves to do this. Just truly heroic. So now I'm going to share what we learned from these studies. I'll start with BDNF since that was our kind of main focus. So what we did was we categorized children based on whether they had intact BDNF, and I'm using the plus symbol to represent that both alleles or genes are present. And then plus minus means one copy is missing. So that's heterozygous or haploinsufficiency for BDNF. And you can see that, across the lifespan that there's a significant difference in body weight, between those with and without BDNF loss. And so if you look at development of childhood obesity, so the yellow bar represents those with loss of BDNF, that 100% of children by age 10 would develop obesity, whereas those who had intact BDNF essentially had the same level of obesity as, just kind of background prevalence of obesity in the United States, which is about 18%. So this, kind of hinted to us that without, nutrition and lifestyle intervention, that there's a great propensity for developing obesity if you have BDNF loss. So when you think about the energy balance equation, there's kind of two parts. There's how much you eat, that's calories in, and how much energy you spend, that's calories out. So we looked at hunger in the patients, and it turns out that the hyperphagia or the hunger score is higher in people with loss of BDNF. So that would hint that it's really an intake of calories that's driving the weight gain. We also did resting energy expenditure, so that's looking at your basal metabolic rate, and we identified that there was no difference. So in other words, there isn't kind of like a slowed metabolism to explain the energy imbalance, but rather it's really energy intake. And so this kind of fits with the mouse studies, because in mice, if you were to take a mouse that has BDNF deletion and you pair feed it, meaning you allow to eat only as much food as its wild type or normal genotype brother, then you can actually prevent the obesity. So by simply, making sure that the right amount of food is consumed, that you can, sort of circumvent the hyperphagia because there isn't like a problem with, resting metabolic rate or energy expenditure. And so it's intake that drives the obesity. And so then that gives a clue for how we might be able to prevent or treat obesity in people with WAGR syndrome. So the other symptom I shared with you about is that mice have decreased sensitivity to pain. So we wanted to look at this in people as well. So we asked the children who were holding this little electrode in their hand that changes temperature, so either really hot temperatures or really cold temperatures, to rate whether or not it caused pain or discomfort. And you can see that the yellow bars, which represents BDNF deletion, you can see the scores are lower than the blue bars, which is intact BDNF. So that means that across all the different temperatures, hot or cold pain is perceived as less painful in people who have BDNF loss. We also looked at cognitive function, and that's through IQ testing, and adaptive behavior, which is using the Vineland. And you can see that there are lower scores for both cognitive function and adaptive behavior if you have BDNF loss. We also looked at social interactions and autism symptoms. And so it turns out that if you kind of divide up the components of the three classic features of autism, which are social impairments, communication impairments, and repetitive behaviors, that it's really the social impairments that seem to kind of stand out in those who have BDNF loss. And because of that, there's a higher rate of meeting criteria for autism spectrum disorder when you use a questionnaire. So the ADI is a questionnaire that is given. What's interesting, though, is if you then have children come in and actually meet with an expert child neuropsychologist, and that is actually done in addition to interviews, it's actually observing. So the O of ADOS stands for observation. And so that's when the child psychologist actually plays with the child and really sees how play and interactions happen. And when they do that, they actually find that really loss of BDNF isn't associated necessarily with full-blown autism, And this seems to suggest then that maybe if you're using just questionnaires to diagnose autism, you may overdiagnose autism spectrum disorder because cognitive and visual impairments may cause some of the same symptoms and kind of be confused for autism. Now, this becomes important in the context of research, meaning that we want to tease out what really is autism versus not. But for families, what we've often told them is that if your child has a diagnosis of autism and it allows for the child to get special services at school and other therapies, by all means, please, hang on to the label, because sometimes the label, even if it's not really capturing the true essence of the diagnosis, it actually can still be beneficial. And so I think that's where we have to kind of tease out the difference between research versus clinical care. So because of these findings of the role of BDNF in both sort of pain, cognitive function, and eating, we now have some ongoing studies. We're looking at how, since everybody has one copy of BDNF left that's normal, it's only one that is deleted, what can we do about the remaining copy? Can we enhance it? Because there's some data to suggest that different types of exercises, different types of diets, and even the timing of meals, so having periods of not eating during the daytime, so not like long periods of fasting, but rather choosing to have eating only when the sun is up and not eating when it's nighttime, can make a difference in how BDNF levels fluctuate in the blood for typical people. And so the question is whether or not when you have one copy lost, can the remaining copy respond in the same way? And so we're kind of looking at that. We also have mice in whom we're testing various different oral medications to induce weight loss. And then right now, the sort of exciting thing that we're working on is, some of you may have heard of the CRISPR-Cas9 technologies that are very exciting for gene therapies. And so we're trying to figure out ways to deliver gene therapy directly into the specific brain regions that control appetite, so that we can get improvements in appetite without causing problems such as inducing excess pain or features that we don't want. So we want to have really specific treatments that don't cause side effects. Okay, so now moving along to some of the other genes, even though BDNF was our focus, we were interested in, kind of opening up, our, palette to look at other things while we were doing the study. So Pac6 haploinsufficiency, or deletion, was already known to have a role in brain and pancreas development. So that part wasn't new. But what we did find that was new and kind of interesting is that when we were doing the brain imaging, we noticed that in children who are healthy and have normal Pac6, they have a normal looking pineal gland in the brain, whereas in people with Pac6 loss, it's not there. You don't see that little bump there. And so this particular part of the brain is important for making melatonin. And you may have heard of melatonin. It's available over the counter, in fact, as a sleep aid. And so people who have smaller pineal glands and who have loss of Pac6, they tend to have greater sleep disturbances. So one of my graduate students who went on to be a professor elsewhere is starting to look at this in her research to try to see if melatonin can be helpful. Another gene that we started looking at was C11 or 46. So this is a gene that is important in chromatin remodeling. So that's in how our DNA is wrapped around different proteins and it controls their expression. And so we found that if you have loss of Pac6, your corpus callosum, so that's the middle part of the brain that connects the two halves, the right and the left, that part is smaller. But it gets even smaller if you also have C11 or 46 deleted. And so I think a picture probably shows this a little bit better. So these are in children and these are in adults. You can see that this is a normal corpus callosum. This is an image of the brain if it were kind of sliced down the middle, in between your eyes, going kind of straight back. And you can see that it looks smaller. This little white band is smaller in Pac6 loss, but if you have Pac6 and C11 ORF, you can see it's much, much thinner. And even the shape of it is starting to change. And so the right and left halves of the brain have to talk to each other. They have to communicate. And this connection between the right and left brain by the corpus callosum is important, particularly for things like auditory processing. So if you have disrupted interhemispheric connectivity, then you can develop auditory processing disorders. What is that? So auditory processing is if, let's say I'm like in a restaurant and it's really noisy and loud, but the person next to me is talking to me, well, I'm able to filter it out. Like I can tell, okay, this is signal is my friend talking. And then background noise is noise, I'm going to filter that out. Or in a classroom, if a teacher is talking, but the kid next to me is like kicking my chair and like making sounds and doing things, can I filter out those annoying sounds and still listen to the teacher? So it's that kind of ability. It's sort of hearing something different in the right and left ear and choosing which one you want to pick. So it turns out that in people who have Pac6 loss, there's actually, that's aniridia by itself, that, there's a very prevalent auditory processing disorder, but there's even more. So double that if you have WAGR syndrome, because people with WAGR syndrome tend to have additional genes deleted, including C11 or 46. So I think that this points to how having additional genes affected can cause symptoms that would be really important to know in terms of helping children in school. So what are some other findings? So going beyond the corpus callosum, we were interested in looking at just all the regions of the brain and to see if there may be, some clues for the different genes on chromosome 11 and how it affects the morphology of the brain or the shape of the brain. So this is surface morphology on the outside and on the inside of the brain, and the different colors are telling you red is that it's a little bit smaller or different from typical brain development, and blue is actually that it's a little bit bigger. So there are like some things that may be bigger or smaller, or just variant compared to in terms of the surface compared to typical developing children. And so I think that that gives us clues for how all the different genes may be affected. So this is ongoing research that we haven't finalized yet, but we're still studying. Some of the things we noticed is that if you look at the adult height, which is plotted on the y-axis here, compared to the size of the deletion on the x-axis, you can see that the bigger your deletion, the smaller your height is going to be as an adult. And in fact, for every sort of 1 million base pairs or 1 mega base deletion increase, you also lose about a centimeter or about a half an inch or so of height. So that means that there may be a lot of different genes along chromosome 11 that are important for stature. And so those are some things that we're also exploring. There were also some patients who had some craniofacial abnormalities, such as a narrow palate and kind of a flattened face and a flattened kind of neck structure, which can put you at risk for obstructive sleep apnea when you're lying down to sleep. And so these are some things that our dental and craniofacial colleagues are interested in kind of following up on in terms of looking at whether or not using dental appliances when children are developing between ages 6 and 12, if wearing like a night guard at night might be able to expand the palate or help with these things to prevent breathing issues or speech issues later on in adolescence and adulthood. We also found that WT1 is very important in development of sex organs. And so basically, it was already known that WT1 has a role in Mullerian duct remnant degeneration as part of normal development. So people start off as female as default. So in other words, if your genes don't turn on and do different things, by default, a baby will be female. And so the only way for you to have a male child, a boy, is for various different genes to turn on during pregnancy that will cause all the female structures to regress and for male structures to develop. And WT1 is one of the genes that's important for that. And I think for us, what was helpful was identifying how this might be important in WAGR syndrome in terms of things to look for in terms of symptoms. So this is a boy. You can tell it's a boy because there's a penis here. You can see in this MRI picture. And what happened was this boy was having recurrent urinary tract infections. And we were trying to figure out why, because, it just didn't quite make sense because boys tend not to get a lot of urinary tract infections, if everything is normal on the inside. So when we looked on the inside, what we found was that here's the bladder. And then behind the bladder was this structure that is exactly where in a female uterus normally would be. And this pouch was actually connected to the same tube that leaves the bladder to make urine. And so what was going on was because the bladder-like structure, which we call Mullerian duct remnant, never went away, there was this extra pouch for urine to back up into and to develop sludge and cause infection and problems. And so, it was advised that, the patient undergo surgery to remove this pouch that was a nidus for infection. So that kind of summarizes the studies results from the phenotype-genotype correlation studies, and we're still doing studies as I mentioned to look for treatments for these conditions. I'm going to shift gears a little bit just to talk about how it was so important for us to work with IWSA to design the study and think about how to make it work and to be able to recruit patients to participate and to stay in the study. And then we had to make revisions to the studies that went along from feedback from families. And it was through the IWSA also that we were able to disseminate findings, kind of in the way that I'm doing now, is to share with you what we learned. So it's like a full circle in terms of communication. So the very first time I met anybody with WAGR syndrome was in 2006. I got to meet Caroline and Irma when they came to visit the NIH, and they brought with them this adorable, colorful rainbow zebra, which my kids love. So it's really special to me, thinking back to our first meeting. And during the years that we worked together, we realized we had to make some adjustments. So for example, some of our testing had this really big lunch buffet that tons of different things for people to choose from to test appetite, but then it turns out that was just really hard for kids to do. So we got feedback from people and switched it over to a single food item test so that kids with visual impairment and being able to feed themselves and reach for things. So that's what was a feedback that was helpful. One thing that was of great concern to us was that we learned that you can have an adverse reaction to propofol, particularly if you have high baseline triglycerides as a risk factor for pancreatitis in of itself, but adding propofol on top of it, because propofol contains a soybean oil emulsion, because if anyone's seen propofol, it's like this white milky liquid, is that it really bumps up the triglycerides. So if you already have a predisposition for pancreatitis due to high triglycerides, this is something that really should not be used. And this is something that we learned, unfortunately, the hard way. So that was one of the most difficult parts of the whole study was when this happened. But my hope is that sometimes when these kinds of things happen, that we can learn in a way that helps other patients. And so now we advise people to check baseline amylase, lipase, and triglycerides before any type of sedation procedure that might involve propofol, and to avoid propofol if there is baseline high triglycerides. We also learned a lot from our rehabilitation consultations while patients were at the NIH. So we had an occupational therapist with low vision expertise. And I would recommend for anyone who's having OT therapies to please ask for somebody with low vision expertise, because you can learn so much from them that just typical OT folks may not have had training on. And so they were able to provide all kinds of adaptive devices that were helpful. And our audiology support staff was incredible in helping, in particular, with the auditory processing disorders. So I'm going to show you a little bit of that. So this was really informative to me. This is called a Paley-Robson contrast chart. So I myself, if I stare at the screen, I can see to here. So I can read the letters all the way down to here. The occupational therapist told me that the typical child with WAGR syndrome or aniridia actually can only see to maybe about here or so at best. And so that was really eye-opening for me because, if I can easily see down here, but someone else can only see there, that's a really different thing. And so they then pointed out that a lot of schools like to use, those light paper, that's a little bit like recycled. It's a little bit gray and it has light blue lines, or they'll use light blue paper with white lines. And you can see, I can see the lines, but if a child who cannot see contrast looks at this, of course they're going to write like all over the place. And so I had a lot of families, tell me, oh, in school, like the handwriting is just completely out of control and it's not, where it needs to be. Well, maybe it's because you can't see the lines, and that's why you're not writing on the lines. And so the advice that the occupational therapist gave was that you should tell all the schools, please only use this kind of really, really bold black and white for people to write on, because otherwise you can't be possibly expected to write on the line if you can't see it. Also, for some patients, they were having significant glare, particularly with corneal concerns and with the aniridia itself, letting a lot of light in, that yellow lenses that were tinted on sort of glasses can be helpful not only for contrast, but also for reducing glare. And so that was something that the occupational therapist shared with us, which I thought was helpful. In terms of increasing self-care and independence, she recommended using these coily shoelaces so that basically you thread them through the holes and then you pull it to tighten and it snaps back because the coil will stop it from pulling all the way through. And so that way you don't have to necessarily tie your shoelaces, but you still have the options of wearing shoes that are not Velcro, because really, in the teenagers or older kids, Velcro options really, limit, the array of shoes you can pick from. And so I think that this allows, for you to pick other shoes, but still be able to tie your own shoes. She also provided these plates that have edges to them and also special grippers. So like it's a spoon that actually Velcros onto around the palm of your hand and has a bent angle to the fork and the spoon. And so then that way it's a lot easier to be able to feed yourself. I don't have a picture of the rocker knife, but she also provided a knife that looked like it was a blunt edge knife that was on a handle so that you could cut food much more easily, because it's really hard to, you know, hold a knife to cut it this way, but if you can rock it, then you can use a rocker knife to be able to cut your food yourself. So that helped. She taught a bunch of kids, you know, how to do this, and they really felt that they were able to achieve more independence in meals because of that. And then our audiology friends showed us these FM devices. So the way this works is the child wears an earpiece and then places a microphone near the teacher. And what happens then is it transmits the voice of the teacher, so it's louder than the background noise. So that helps with the signal to noise ratio. Also, it's really important for the kids to be at the front of the classroom, so that way they can have a little bit more focused attention and also kind of avoid, you know, how the back of the classroom can get a little noisier and a little more disruptive. And so trying to avoid that background noise can help with the auditory processing to be able to learn. Of course, now with, you know, COVID, you know, I think one of the things that we're sort of realizing is, at least when my kids were doing sort of Zoom classes, is that they don't mute all the kids and everyone's talking. It is, you know, no one can hear anything. And so I think that giving feedback to teachers to make sure to only have the one person who's teaching on and everyone else needs to be muted is really critical, because otherwise there's just no way to focus. I mean, I think all of us would struggle with that. So we learned a lot from this. You know, we learned that family perspectives are really important. For example, we started to hear families talking about how a lot of their kids seem to be, you know, kind of experiencing clumsiness because they would like bump into objects, especially on their sides, at their peripheries. And, you know, they would repeatedly express this to, you know, care providers and be told, oh, well, it's just visual impairments or maybe neurocognitive deficits. But then when you like ask a little more specifically, you hear from the kids that it's because they have double vision unless they turn their head to the side. So that actually sounds like something a little more than just overall vision decrease. And so what we found out was that some kids actually, when you ask them to gaze, so in this young man, we asked him to please gaze this way, and you can see that this eye moves toward the, you know, center like it's supposed to, but this eye got stuck. It never went all the way to the edge. So there was a problem with lateral gaze or sideways gazing. And what can cause this? Well, the cranial nerve 6 is the one that's responsible for this. It's called the abducens nerve. And so the one on this side here looks perfectly fine. It's right there like it's supposed to be, and that's why this eye moves just fine. But this one, you don't see any little black dot to represent where that nerve should be. And so in the absence of that, that's why the eye wasn't able to move all the way over. So this is really important to know, because now we understand it's not clumsiness. It's actually a visual impairment. And so it requires, you know, understanding that, you know, the periphery, you know, can be dangerous unless you're cautious to make sure that we don't, you know, have accidents or problems because we don't look to the side. So it is an important awareness to have. The other thing we learned, and this is alluding back to the autism diagnosis thing I was mentioning before, is that what researchers want, those priorities, may not always be the same as what families want. And we've got to align this by talking, because maybe the researchers are like really, really focused on trying to like be really precise about the diagnosis of autism-like features versus true autism. So our neuropsychologists were writing this out in gory detail in their notes. And then we had to realize that we had to be careful, because these are research notes. And if this gets back into the regular medical chart of the child back home, it's possible that you might actually lose social services if you don't have an autism label. So we actually revised the way we do things, and we started to seclude, you know, what was research and what was clinical. And so we changed it. So we actually had our neuropsychologists try to like start to word things differently, like would benefit from services because of these autism features and things like this. And that's incredibly important, because we obviously don't want anyone to, you know, to have reduced services because of a diagnosis being removed. So this is why communication is really important. And that's why, you know, having kind of a clear distinction between what's clinical and what's research is also, you know, just a really important part of research. And then this is my last slide. It's basically to show that we learned that since WAGR syndrome affects essentially every organ system of the body from head to toe, that nobody can be, you know, an expert at everything. So we had to collaborate with so many other experts, and we had to collaborate with families, because ultimately the real experts are the parents. It's the parents of children who can share with us symptoms and things that we never even thought to ask about. And so this is why collaboration is really, really the only way, or the way of the future, really, to make a difference in promoting medical advancement. And so along those lines, I have to say thank you to all of the research team and collaborators who made the studies possible and are continuing to collaborate with me to this day. And then an extra special thank you to the families who joined our research studies. And then for anyone who would like the link to the 60-minute presentation that was done in 2018, I updated a little bit for the 2020 data. This talk was just extracted from that to make it shorter, but if you want to hear the whole thing, it's here. And it will also be available on the IWSA website. So thank you very much, and I'll be happy to answer any questions.